student talk, and it's like after a very long time a student is ready for a talk in Curie Club, and it's like before we had some 7 or 8 student talks, and all from like Asif who gave on NMR, Deepansh who gave on Fortran, one I gave on to build an NMR, and Gurpreet who gave on how to visualize a molecule in lab, and V who gave on NSE, and Manish who gave on natural product synthesis, Srijit who gave on liquid crystals, then Abhinay who gave on UV visible instrumentation. And now in that series, I think after a very long time, we have with us Shwetha, who did her schooling from National Public School, Bangalore. Now she is in chemistry majors, 3rd year. She was elected for Asian Science Camp last year, and this time she was even elected for this Lindau Nobel Laureate Meeting, which she will probably be talking in her talk. And in summers, this time she worked with Hanudatta is Atreya, right? Okay, in NMR Center in IISER Bangalore on protein NMR. And so NMR, it's basically long, maybe 90s, was started by Stern, Gerlach, and all, these people in a series. Then it was I. Ravi, who even got Nobel. And then it was Felix Bloch and E.M. Purcell, who for the first time got an NMR signal. And these both people were awarded Nobel. And then it was Richard R. Entz, and who got Nobel for 2D NMR and Fourier transform NMR. And then it was put with Rich, and it's a very odd field. And NMR, is nowadays employed in many chemical systems and biological systems. And NMR involves, quantum mechanics and spectroscopy, which are made for each other. And I hope now Shwetha should talk about it. Let's welcome Shwetha.
Thank you so much, Pradeep. So, like Pradeep said, this summer I worked on NMR spectroscopy. So basically, I used NMR spectroscopy to study proteins. The main advantage of this spectroscopy is the fact that it can give you atomic resolution. You can get information about each and every nuclei which are NMR active in your system. So we'll first start off with what is spectroscopy. So spectroscopy is basically the interaction. Spectroscopy is basically the interaction of electromagnetic waves with matter. And so we all know that electromagnetic waves have 2 components. The first, the electrical field and the magnetic field. So when there's an interaction between the electric component of the electromagnetic wave with matter, it gives rise to spectroscopy such as UV, IR, and microwave, which are in this region of the electromagnetic spectrum. This is the entire electromagnetic spectrum. And when there's interaction of these magnetic fields with, when there's interaction of the magnetic component of the electromagnetic wave with matter, it gives rise to phenomena known as NMR and EPR. So in this talk, I will be talking to you about nuclear magnetic resonance. So every fundamental quantity has an intrinsic property which is called as spin, and this is generally specified by a spin quantum number I. So now this I can be either a positive integer or a half integer or even 0. And the examples of nuclei where I takes these various values are written here. When I takes the value 0, the examples are the 12C or the 16 oxygen. When I becomes a half integer, the examples are proton, 13C or 15N. And when I is greater than half, the examples are 2D, and this is the isotope of hydrogen, the deuterium, and 14N. So associated with this quantum number I are 2i plus 1 degenerate states, which run all the way from minus 2i plus 1 to 2i plus 1. So now if we take the example of I is equal to half, that is an example for any of these spins, 1n, 13C or 15n, and you put this value half in this equation over here, we will get 2 into half plus 1, which is equal to 2 states. So this gives us to 2 energy states. So now, like I've already said, these two energy states are degenerate. But when a magnetic field is applied, and this magnetic field is generally applied along the z-axis, so this leads to the separation between these two energy states. So now, so like I told you, every fundamental property, every fundamental particle has an intrinsic property which is known as spin. So then, when this spin is introduced in the magnetic field, which is along the z, it experiences a torque. Now because of this torque, it starts precessing. So this is my magnetic field. This is along the z. And now I told you that every fundamental particle has an intrinsic property called spin. So now when this spin is introduced in this magnetic field, it starts precessing. What do I mean by precessing? It starts going around talk about a little later. So an analog of this is that, I'm sure most of you or all of you must have seen a gyroscope. So a gyroscope, when you just place it on the ground, it tilts towards gravity, due to the force of gravity. Now the same gyroscope, you take it and if you spin it and then you place it on the ground, it starts processing due to the torque which has been applied on this gyroscope by the gravity. So it starts spinning across, it starts spinning about the torque which is applied by the gravity. So similarly here, the spin over here will start processing about the direction of the magnetic field. So now, so this like we just discussed, for a single spin, for a spin half system, there are two energy states. These are known as the plus half and the minus half. So the plus half states are those in which the spin is processing in the direction of the magnetic field, and the minus half are those is that state where the spin is processing in the direction opposite to that of the magnetic field. So I have plus Z and I have spins which are processing like this, and I have the minus Z where the spins are processing like this, the direction opposite to that of the magnetic field. So now this energy separation, this comes out to be gamma h beta naught. And so what exactly is this gamma? So this gamma is known as the gyromagnetic ratio. So now every nuclei has a fixed number. This gyromagnetic ratio is a fixed number for every nuclei, and the energy separation depends on this value of gamma. Okay, so then and it is a known fact that the gyromagnetic ratio for hydrogen is 4 times that of carbon. So which just means that in case of hydrogen, the difference between these energy levels will be 4 times as what it is for carbon. And another important thing over here is that this gamma b is defined, omega is defined to be minus gamma b, and this omega has a nano frequency. So I told you all that when a magnetic field is applied, the nuclei starts processing about this magnetic field. So the frequency of this precession is known as nano frequency, which is defined to be as minus gamma b, minus gamma b. So this is a NMR spectrometer. So the NMR spectrometer has 3 major components. So the first one is the magnet. The magnet is superconducting, and the magnet is immersed in liquid helium. Now liquid helium has a boiling point of about 4 Kelvin. So the main point of using liquid helium is so that we maintain a temperature of less than 4 Kelvin, which is necessary for the magnet to retain its property of superconductivity. So surrounding this liquid helium is liquid nitrogen. Now liquid nitrogen is very well known that it has a boiling point of about 77 Kelvin. So why do we use liquid nitrogen? It's so that we don't lose liquid helium by evaporation. The rate of the liquid helium has a boiling point of 4 Kelvin, whereas the liquid nitrogen has a boiling point of 77 Kelvin. So basically, to save liquid helium, we use liquid nitrogen. Liquid helium is very expensive. It's about 1,000 rupees per liter or so, and it's not very easily available. So because of that, we use liquid nitrogen. The second important thing here is this is known as the RF power generator. So what exactly is the RF? I will tell you a little later towards my talk. So this is a very important component of the spectrometer, and the third thing is the console. So this controls your entire spectrometer. It's like maybe a brain of a human being, or the CPU of the system. This controls the entire working of this NMR spectrometer. Okay, so now we come to one of the most important and basic things in NMR. So I'll tell you now about the 1D NMR. So like I told you, for a spin half system, I told you that there are 2 energy levels. This one corresponds to plus half, and this one corresponds to minus half. So then since this is lower in energy, it is a known fact that there will be more population in this energy state. So let us say that our sample has 100 spins. Let's take an example where our sample has 100 spins, and all of them have the spin quantum number as half. So because we've taken it that way. Since this state is lower in energy, let's say that this has about 60 spins and this has about 40 spins. So and I also told you people that the spins which are in this state, they are processing about the positive z-axis. So these are processing this way about the positive z-axis, and the ones which are here, they are processing in the opposite direction. So now if we see the net difference between this is about 20 spins. So these are processing about the positive z, and these are processing about the negative z. So there is a difference of these 20 spins. So from this we can come to know that the net magnetization of the sample is along the positive z, because there are more spins which are processing about the positive z. So this is something which is very important to understand. energy levels to split, and my net magnetization is about the Z, about the positive Z. So next what I do is I apply a 90-degree pulse. Okay, now what basically this 90-degree pulse does is that my net magnetization is about Z. Now I want to bring this to the transverse plane. Why do I want to bring this to the transverse plane? Because if I make my measurements along the Z axis, I have this giant magnetic field. Now this giant magnetic field has the magnitude of the magnetic field here is several Tesla, several magnitudes higher than that of the magnetization which is produced by these nuclear spins. So because of that, I'm going to bring the net magnetization from the Z to the transverse plane. So now how do I do this? This is done by applying an RF pulse. So RF pulse is basically it's an oscillating magnetic field. So I told you that these spins are spinning with a particular frequency, and this frequency is a Raman frequency. So I'm going to apply an RF pulse, which is depicted by the 90-degree here. So that what it will do is that it will bring this IZ, it will flip this by an angle of 90. So when I apply it along X, this will flip to Y, and it comes down to the transverse plane. So this RF pulse is basically an oscillating magnetic field whose frequency is the same as that of the Raman frequency. Till now, if you understand so much about the RF pulse, it's sufficient. We'll get into more details later on. So now I've applied this RF pulse. Initially my magnetization was along Z, and I've dropped it to the Y. So now the RF pulse is removed. This RF pulse is a magnetic field which is applied about the X. So the RF field is removed. So now there's again this bulk magnetization. So what is the property of this nuclei? We start processing about that bulk magnetization. So when it processes, what is the frequency with which it's processing? The Raman frequency. So it is this Raman frequency which we catch in our spectrum. So this starts processing, and this starts processing, and now I have coils which are under transverse magnetization. I told you we can't keep coils in the Z axis because the giant magnetic field has a very huge value, and we want to catch the magnetization of these pins. So we keep it on the transverse axis. So now this is oscillating magnetic field, and according to Lenz's law, this oscillating magnetic field will induce a current in the coil, and it is that current which is measured. So what is the current? It is basically the frequency of this precession, which is the Raman frequency, and which is the information we get in the spectrum. So now another important thing here is that this signal which we are measuring is known as FID, which stands for free induction decay. So why is it called a decay? It's because since this spin is processing about the Z axis, and along with this precession, every system, it's a property of every system to come back to its equilibrium. So along with this precession, this spin it wants to come back to the Z. So now I have coils only in the X axis. So this is processing, it's catching the Raman frequency, but along with this precession, it's coming back to the Z because of the property of the system to come back to equilibrium, because of which there is a decay. And it's known as induction because of Lenz's law that the oscillating magnetic field it induces a current to the coil. So I'll just repeat this again. So basically what we've done, we have the magnetization about Z, we've applied an RF pulse, and the RF pulse has done something, let's say, and it has brought this magnetization to the XY plane, and now the nuclei, the spins again start processing about the Z, and then there are coils which I have in the transverse axis. So these oscillating magnetic field, they induce a current in the coil, and this is what comes in the spectrum. And now this system has a property to come back to its equilibrium. So it comes back to its equilibrium because of which we see the decay signal. So once we get the signal, so the signal is obtained in the time domain. So that's the T1 here. But if you notice in an NMR spectrum, you actually see the values in ppm, parts per million, but that can be converted to the frequency scale. So what we basically see is the Raman frequency. So we have to convert this time domain to the frequency domain, and this is done by Fourier transformation. So we have time domain, and we convert by Fourier transforming it, we convert it to the frequency. Okay, now so in this slide we've discussed that since gamma for every nuclei is different, the Raman frequency of every nuclei will be different. So by this we are able to differentiate between different nuclei. I can differentiate between a carbon and a hydrogen because the frequency with which they will be processing is different. But is it possible for me to differentiate between different types of the same nuclei? So basically, this can be done by using the property of chemical shift. So we all know that the nuclei are surrounded by electrons, and these electrons shield these nuclei from the external field. But these electrons which are going around these nuclei, they also induce another magnetic field, and this magnetic field is in the direction opposite to that of the magnetic field with which the nuclei are processing. So now, because of this, now see here, let's take an example. This is the most common example taken. So we have the example of ethanol, CH3CH2OH. Okay, so now we are recording for proton. We are recording the NMR for proton. So we have three hydrogens here. One belonging to this, one belonging to the CH2 group, and one belonging to the CH3 group. Now, all of us know that oxygen is an electronegative atom. So what it has a tendency to pull all the electrons away from hydrogen. Now, if you see in this molecule, all these hydrogen, they experience a different field around them. Because the electron density around each of these hydrogens are different. Now, since the electron density around each of these hydrogens are different, they experience a different, the field experienced by each of these nuclei is different. And since this field is different, all of them they appear in the NMR spectrum at different ppm values or different frequency values. So by this property, we can even differentiate between nuclei of the same type. Like we can differentiate, like here we are able to differentiate between the different hydrogens because of the different electron density which they have around them. So this is where NMR is able to provide you atomic level information. If there is a carbon and a hydrogen, due to the difference in the diamagnetic value, I can tell you that this is a carbon and this is a hydrogen. And by using the property of chemical shift, I'm even able to differentiate between the different hydrogens which are there in my system. Okay, yeah. So this is another common concept which is known as coupling. So there's a thing which I forgot to tell you earlier. So here we see that in this case, we got this to be the peak of CH3, and this to be of CH2, and this to be of OH. So since this arises due to three hydrogens, the intensity of this peak is greater than this, which arises due to two hydrogens, and the intensity of that is greater than this, which arises due to one hydrogen. So now, as far as it's a simple molecule like ethanol, it's okay. I mean, we are able to assign the peaks that this belongs to the hydroxyl hydrogen, this belongs to the CH2, this belongs to the CH3. But if my spectrum is very complicated, then the spectrum is going to look very crowded. So then, in that case, what scientists do is that they decouple a spectrum. Okay, so we are not going to worry about how decoupling is done, but what basically the decoupling does is that it makes that now this peak is split up into three. I'll tell you how the splitting occurs. But what decoupling does is that it makes that all these three peaks, it collapses all these peaks together, and you get a single peak. Now, what is the advantage of this? Your spectra is going to look less crowded, and your intensity of these peaks are going to rise because the three peaks are now joining together and becoming one peak. The same thing happens here, and the same thing happens over here. So your intensity improves, and your spectra looks, your spectra looks a little less complicated. Now, how does this splitting occur? So again, let's take an example. Suppose I have this hydrogen, and say there is this hydrogen. Okay. So now this hydrogen basically. We've discussed the two energy states for a single state mass. So the nuclei over here can either be in the alpha state or in the beta state. So the spin can either be up or the spin can be down. So due to this hydrogen is split up into two. Now the same way if I have H2, as in if I have two hydrogens over here, so then this hydrogen will be split up into three. How does this follow? If this proton has n neighbors, it will be split up into n plus 1 peaks. Okay, so if it has two, then this spin can either be in the up state or in the down state, because of which we see a doublet. In the same case, if this proton has two neighbors, it will be split up into three. So that's the reason why over here. So this is ethanol. So this CH2, it has a carbon which has three hydrogens. So it has three neighbors. So that is, if there are n neighbors, we see n plus 1 peaks. So since there are three neighbors, we're supposed to see 4 peaks. So that's why in this case, this hydrogen has three neighbors. So then that's why in this case it has three neighbors. So it's supposed to split up into 4 peaks. That's why we see 4 peaks over here. This is a quartet. So similarly, this hydrogen, it is connected to a carbon which has two hydrogens. So it has two neighbors. So it's supposed to split up into 3 peaks. Okay. So whenever you're recording an NMR for a chemical molecule, like for any simple chemical molecule, these spectra is okay. You can assign your spectra. You can get the value of the gamma frequency. That is the chemical shifts. But when you come to recording data for a protein, the spectra gets very complicated. You cannot get any information from the spectra. You possibly, like the regions which have been marked, I can just know that in my protein there's a C alpha which is present, there's a methyl region, there's a amine region. That's all I can get. I cannot get information about the chemical shifts or the coupling, or I don't know whether they're coupled, they're decoupled. I have no information regarding the spectra. So in this case, so it was during this time that people realized that we need another dimension to do the measurement. So that's when 2D NMR was introduced. So how exactly we record data? So this is the first block is known as a preparation block. So basically all these blocks, they have these pulse sequences. We've discussed just one, the simple 90-degree pulse which rotates in net magnetization from IZ to IX. But all these blocks here, they may have a simple set of pulse sequences or a complicated set of pulse sequences. So we're not going to worry about that. So what this preparation pulse does is, initially at equilibrium, we know that the number of spins in this state is going to be higher than that state. So my net magnetization is going to be about Z. So this preparation pulse can be as simple as just bringing this from the Z to the XY, or can be a set of complicated pulses. Next is known as a period of evolution, which I will just discuss about. And again, mixing is another block where it can again be a set of simple pulses or complicated pulses, and it depends on what data I'm looking for. Whenever I'm recording a biomolecule or a chemical molecule, it depends on what data I'm looking for. So basically, how this works is, so in the first case, this is my preparation, and I set my T1 which is written over here, over evolution, I set that to 0. So T1 is 0, then immediately there is mixing, and then there is, here this is detection. So this detection is like a simple 1D which we discussed, the free induction decay. So for T1 equal to 0, I record different data types, which is the T2. Now next what I do, I implement this T1 to say T1 now becomes delta 1. So when my T1 is equal to delta 1, I run the entire experiment again. So I'm going to get again another series of points. So next, this is done many times. So we can say 2 delta 1, and I record this again. So this is done for many values of T3. So now if you see, we are recording along the T2 dimension, and we are recording along the T1 dimension. So then similarly, in 1D now, how we discussed that we have this T1, I do a Fourier transformation, I take a frequency. So similarly over here, when I do a Fourier transformation from, I do the Fourier transformation of T1 to T2. I get omega 1 from omega. So now I've got information in an extra dimension. So now we'll see how this helps us to study larger molecules. Okay, so this is a pulse sequence of a very useful, it's a very useful experiment, the HSQC pulse sequence, and it's used by any person who does protein NMR. I mean, this is the first set of experiments which you will do, and I'll come back to this pulse sequence a little later. So this is an HSQC spectra. So this is an HSQC spectra of a peptide which I was working on during my summer. So the peptide is known as an insulin growth factor like binding protein, and it's used for cancer therapeutics. So I was given this peptide, and firstly why is this HSQC done? So then this, as you can see, this is a polypeptide chain. Okay, it's basically here what is depicted is a dipeptide. So what information this gives you is, whenever I do a 1D experiment, for one frequency coordinate, I have intensity, correct? Like I get the intensity for one frequency coordinate. But in a 2D experiment, what I do is I plot intensity versus two frequency coordinates. And the important 2D experiments are those in which there is a correlation between these two frequency coordinates. So now in this polypeptide chain, we've already discussed that 2D gives us information in 2 dimensions. So now we have over here, we see that this hydrogen is coupled to this nitrogen. So the HSQC which I am doing over here is the HN HSQC. So along this dimension, I have the Raman frequencies of the hydrogen which are present in my protein, and along this dimension I have the Raman frequencies of the nitrogen which are present in my protein. So now if I find a peak over here, what this tells me is that like now my protein is made up of many amino acids. Let's just take an example. Say valine, alanine, leucine, glutamine. So it's made up of many amino acids. So now each of these peaks, they correspond to an amino acid. So what I'm supposed to, I get this spectrum, I'm supposed to assign a spectrum. As in I'm supposed to make out what all amino acids are there in my spectrum, because that is something which I do not know. So what I do is that now if I get a peak over here, that means the Lambda frequency of hydrogen, which corresponds to this x-axis, and the Lambda frequency of the nitrogen, which corresponds to this y-axis, it tells me that these two are coupled. Coupled as in they are joined together by a bond, by a single bond. So then I go, there's something known as a NMR biological resonance bank. So that has the Lambda frequency values for all the atoms which are present in the amino acid. So now I know I'm getting a peak over here which corresponds to a certain Lambda frequency of hydrogen and a certain Lambda frequency of nitrogen. So then I see this value from here and then I go back to that data bank. And then that data bank I look at which amino acid has this particular value for its nitrogen and hydrogen. And once I get to know this, I know that this belongs to gluten. So similarly, I do for all this. So what is the importance of an HSQC spectrum? Basically, it tells me the different amino acids which are there in my protein. So another important experiment which is done with HSQC is now I have a protein structure. So I have a protein and I want to find out the structure of the protein. I want to know that which are the amino acids which are exposed to the solvent. So if in my solvent, suppose I have taken my solvent to be deuterium. Okay, I have taken D2O as my solvent. So but we are recording a hydrogen and nitrogen. So then what happens is that the amino acids which are there, which are exposed to the solvent, the hydrogen in those amino acids, this hydrogen, they will undergo exchange with the deuterium which is there over here. So now once these hydrogens are replaced with deuterium, there is no more coupling between the nitrogen and the hydrogen because the hydrogen is lost in the solution. So because of which I will not see that particular peak because the hydrogen has been replaced by deuterium. So I am not going to see that particular peak. So